the round table discussion. So we had really the great chance to have actually five people to do that. So I mean, Florian already was presented, also you, Gianluca as well. So I will also present Dr. Russell Kraft, Dr. Stephanie GUERET, and Stefan LAUFER. So I will start with Stephanie. So after studying organic chemistry at the University Claude Bernard in Lyon, France, Stephanie completed her PhD at the University of Auckland in natural product synthesis, and then she moved to Novartis for a postdoc. After postdoctoral studies, she took a senior research scientist position at AstraZeneca to join the AstraZeneca Max Planck Institute satellite unit. And together with a small team of students and scientists, she led major projects of the satellite unit. And after that, in 2019, she moved to Gothenburg, Sweden, to work on small molecule projects for liver disease, from hit to lead optimization. Recently, she was promoted to associate principal scientist, focusing on small molecule medicinal chemistry in the field of cardiovascular, renal, and metabolism diseases. Her current research interests in synthetic and medicinal chemistry, with broadening towards chemical biology to solve key questions on how to move forward novel chemical entities towards drug-like compounds. So welcome, Stephanie. So then Dr. Russell Kraft completed his degree in chemistry at the University of Sussex and subsequently obtained a PhD at the same university under the supervision of Professor Phil Person, working on a radical cascade approach to the photosynthesis of alpha cyclopropanoic acid. After a brief hiatus from chemistry in the construction industry, he worked at Evotec as senior scientist, and is currently employed at Symeres as a project leader in drug discovery department. Russell is also a member of the FMC Best Practice Working Group. And finally, Professor Stefan LAUFER is a professor and chairman from the Pharmaceutical Medicinal Chemistry at Tübingen University. He received his degrees from Heidelberg University, and after 10 years in pharmaceutical industry, he joined in 1999 Tübingen University as chairman for med chemistry. His research interests are anti-inflammatory and cancer drug discovery, and this compound from his lab enter clinical development phases, and Professor LAUFER chairs the IFA, so Interfaculty Center for Pharmacogenomics and Drug Research. And as part of his work, his proprietary kinase inhibitor collection is established. He's associate editor of the ACS Journal of Medicinal Chemistry, authors of more than 500 publications, 15 books, book chapters, and inventor in 42 patent families. So thank you very much, guys, for your time. And so then we did the first question from one of you. So could you comment on the different challenges that chemists encounter when developing a drug or chemical probes, and are the different parameters that might be considered? So maybe one of you, like, I don't know, Gianluca, for example, can answer this question.
Yeah, well, actually, I have to say that maybe the most obvious thing could be the access to different techniques when you need to validate probes. But this is not a real problem in my opinion, because I'm pretty well convinced that we were able to show that collaboration is the key. So companies are quite open to collaboration. CRO, well, there are a lot of CROs that are able to provide services in this regard, even to academic groups that are not able to have a direct access or accessing collaboration with this kind of essays. But in general, the other difference could be, well, the strategy. So you mentioned before DNA encoded library, or let's say fragment-based discovery, in particular if you consider the X-ray based fragment discovery. Of course, it's something that could be not so immediately accessible to academia, but again, collaboration is the key, in my opinion.
Mm-hmm. Thank you very much. Maybe Stephanie, you want to comment on this?
Not really comment on what Gianluca said. I agree that collaboration is the great way to make sure that you reach all the aspects of your chemical probe. But maybe one thing, if you, for example, you have a target which does not contain any structural information, that would be a great challenge, either find your starting point and to develop the HTS or medium throughput assay that will allow you to get the starting point for your probe, and also the lack of structural information might make a little bit more difficult to find your anchoring point to attach your linker in order to label your probe and then follow on with the assay you need to validate the probe. So this is one, I think, example, but there is many challenges.
Florian, if you want to add something more?
Just to jump on what Stephanie mentioned, and I hope this is making the difference, at least between pharma and academia. You don't have to forget the PK profile, meaning even the in vitro PK profile. You go in cell line, you have clearances. Okay, to validate a compound on a test which is 20 minutes, it's no problem. But then if you really want to validate your target, and I mean biologically speaking, validating your target, in vivo veritas. So you need a compound which is stable, at least half an hour, which is not killing all your organs, which is not inhibiting all your liver and all these kind of parameters. Unfortunately, all these tests cost a dramatic price.
Sure.
And I think this is where I am fully with Gianluca. Meaning, if you are aligned on a target between an academic group and a company, then let's join forces, because this is where a private company can help for sure, having all this panel of assays in place, and where all the expertise of academic here is because of creativity, innovation, and of course all the academic tools they could have access in their network.
I fully agree with you on that. So then the next question is like, so we know that the drug discovery process is a very long path. And how long does it take usually to generate a chemical probe? So maybe Russell, you want to comment on this?
Yeah, it's an interesting question. I suppose the real question here is like, how long does it take to validate a probe so it becomes a high quality probe? And I think sort of some of the initiatives that have been discussed so far, like the Structural Genomics Consortium, help sort of accelerate that process by making these probes available to the community, so these can be tested. Yeah, because only with the high quality probe can you sort of generate this worthwhile biological data that can be appropriately interpreted. So, okay.
Maybe Florian, if you want, yeah.
I would say 2 years, meaning average for us at BI to have again the in vivo ready tool. It's at least 2 years.
Well, again, in my opinion, of course, I agree, but in general, it depends on how many resources, human resources you put on it, not just economic resources. So just to make my example of the story I just told, being a rather small academic group, actually, well, we started that project in 2013, and we published it 4 years later. And it wasn't the problem of publication. We just finished validating everything 4 years later. But of course, if you double human resources and double the efforts, double the collaborations, well, it could be reasonable to have it in a couple of years.
Stefan, maybe if you want to add?
I think critical in this issue is not only how many resources you have, but especially how many assays you can run in parallel. Especially in academia, you have in your lab probably, if you're lucky, 5 or 6 different things. But for the probe, you need at least between 10 and 20. And it's naive to think that you can run 20 collaborations in parallel. If your lab is specialized in synthesis, it's not a good idea to focus on probes and to do 20 collaborations. You should have at least in your lab basic characterization, which is at least binding, inhibition, or whatever. You need some metabolic stability, you need at least an orthogonal assay and some basic physicochemical properties. Without this having in your lab, it's very difficult to make in a reasonable time a medium quality probe. So I would argue 2 years is minimum, 4 years in academia is more the rule than the exemption. So it's nothing to make quick and dirty.
From our experience in the satellite unit, it also depends a lot on which target you go for, and does the biological assay are known, or do you also need to develop your biological assay and your cellular assay, including developing cell line and things like this? This can lengthen your discovery process quite a lot, like of 2 years just for the biological assay sometime.
Thank you very much for all this information. So I have a third question. So most of the marketed drugs are not good chemical probes for mechanistic investigation studies. So for example, they are not selective enough. So could you comment on that and advise the audience and on how to discard poor quality chemical probes? Does anyone want to start?
Well, I could say just follow the webinar given by the best practices working group, actually, and see all the hints and suggestions we gave. Seriously speaking, but well, actually, I wasn't joking at all. Experts can provide very important suggestions in this regard, and of course, people in Boehringer, SGC, chemical probe portal, and so on. Are a very useful starting point, in my opinion.
Anyone want to add something else?
Well, as being a synthetic chemist, I suppose the first thing I do is to sort of have a look at the chemical structure of the probe. Does the structure raise any flags? Are there any sort of reactive groups, any Michael acceptors, things like this? Does it hit any Payne's filters? But you have to be a little bit careful with that, because sometimes good molecules can be discarded by that. So if those sort of things were there, I'd ring alarm bells. I'd also maybe have a look to see if there was any closely related structures or similar core scaffolds, see whether there was any related biological info on those. If there was, again, there have been questions about the quality of the probe. So that's one approach.
Oh yeah, if you want to.
Yeah, so probably I'm very biased by the fact that I am with a lot of biologists around me, but the potency on the target and the selectivity on the target would really make the difference. Of course, you have to look at what you are targeting. Is it an in vivo or in vitro application? If it's in vitro, you can tolerate, of course, not a great solubility. You can tolerate instability. So I think it's really based on what you want to achieve. It's to validate a pathway. If it's to validate a target in vitro, you can be much more flexible. Of course, if you want to go in vivo, here you will start to be in trouble. But I think, as mentioned by Stefan just before, you need to have the basic phys-chem properties information, and here you need to have this information. Meaning, if you have an intracellular target and you have no penetration into cell lines, forget it. You will never validate your target. So I think you can put on the table rules, no problem, but every tool will be adapted to your target.
Stefan, if you want to add.
Yeah, a very often made mistake considering what is a probe and what not is the quality of the journal in which the so-called probe was published first. It doesn't matter if the compound was published in Angewandte, Nature, or Cell, or Science. It's important what are the properties of the probe. And very often, in very early stages, compounds with poor characterization are published in Science or Nature, because this is a front-runner paper. And then all the world is using this compound as a probe because it was heavily cited and used so often and initially published in Science. So please don't make this mistake. It's exactly as Gianluca said, there is really a good because it was made by us. Serious, what is a probe? And this question especially we answer, I think, in the first 3 slides. What makes a good probe and how to get the idea where to find good probes. And really, it doesn't matter the quality of the first publication.
Well, it doesn't matter on the quality of later publications as well, because even if a probe's been proven to be bad and there's lots of publications demonstrating that, you'll still find publications later on, people using that probe. Staurosporine is the sort of archetypal example here, I'd say.
Absolutely. All right, so then one last question. So what are for you the must assays to validate a chemical probe?
Well, this is a $1 million question, actually. Well, it strongly depends again. Well, in my opinion, for sure, you need to be sure that you are engaging the target. So you need biophysical and biochemical assay. In particular, you need orthogonal assays in terms that you are not observing another effect that could be affecting your results without you understand this. So just in principle, if you are using, let's say, a thermal shift, could be a good starting point for screening quite a number of it, but of course, it's something that you cannot use for validation. So you need ITC, or you need SPR, and also you need biochemical assays, in particular in cell assays, if you want to gauge and assess the in-cell target engagement, just to start.
Florian, do you want to add?
For me, which is really mandatory, and I do not have any chairs in the company, so I will continue to nominate Eurofin as a kind of benchmark, because this is the one which is the most available, and also for academic, you can get quite good stuff there. But you need to be selective, meaning we have thousands of papers showing that a great compound is super active in your target or in your mode of action, because it's not touching your target, but another one. So you need to be selective. And even if it's in vitro, meaning if you go in cells, you could have many off-site targets, and you need to be sure. You know, if you have a basic amine with a log D of 3, be sure that you are GPCR clean before to do anything with this probe compound, because otherwise you don't really know if it's coming from your target.
Stefan?
Yeah, and I think, as it's also heavily mentioned in the EFMC best practice, is the target engagement. You need to demonstrate target engagement, both in biophysical and also in cell target engagement, to be sure that the physiological effect that you see is really linked to the target you want to inhibit or to modulate, because the probe is supposed to only act on this target. It's not a drug which is supposed to do a physiological effect at the end, the right one. A chemical probe is to address a protein of interest and only this one. So with the selectivity, you need to demonstrate the target engagement.
Suppose another way is to use some complementary biological assays. So to validate the functional response to a chemical probe, so to sort of a mutation that removes this compound binding, or some sort of where the biochemical target has been removed through sort of CRISPR-Cas9 sort of approaches, to really show that whether that probe is interacting with that target.
Stefan.
So at least for kinase inhibitors, which are very often difficult probes because old ones are never selective, the SGC has made a nice checklist of roughly 10 bullet points. If you allow me to share the screen, I can show this checklist, and this is available in the internet at the SGC site. And this can be easily also used for, let's say, other probes, not one-to-one, but can easily be adapted. So would you allow to share my screen?
Yeah, sure, absolutely.
Okay. Okay, do you see now this word table?
Yeah.
Okay. Yep. So this is the SGC checklist for kinase inhibitors. And this is very prototypic and can be easily adapted for other compound classes as well. And gives you a nice hint what should be known about the compound. Okay, and this, as said, is on the SGC side, can be freely used by anybody.
Yeah.
Okay.
Very nice. Thank you very much. All right. Anybody want to comment or add something?
Well, maybe just another point that could be added is the fact that, again, in my opinion, if you are targeting, let's say, a protein, whatever is the target, there is still the importance of having different structure. So different probes with different structure could be extremely important because, well, you can have a different way of interaction, and also you can observe different, let's say, effect considering the interaction with other partners. So having different chemotypes could be quite useful and something, well, very important for the scientific community.
And to follow up on this also, it's not always possible, but optimally you should also get closely chemically structure, which is a negative control, which will have no effect.
Sure.
Yeah.
Perfect. Thank you very much. So then I think we can stop here.